And it's very nice to speak to you in person. We've been having a few discussions last time, and we've been working together now for maybe a year, I guess, maybe a little over a year. And I'm very excited about it because it opens a new avenue for diagnostics in metabolic diseases. And I think it's better that you tell me a bit about what you are working on and I've been part of.
Okay.
So could you tell me a bit about yourself and what you're working on?
Okay. So my name is Esther Sammler. I'm a clinician scientist. I'm originally from Germany, but I'm here at the moment at 10 o'clock at night in my office at the University of Dundee. So I studied medicine and graduated from the University of Ludwig Maximilian University in Munich. Then I started my training as a doctor in neurology at the University of Heidelberg and eventually moved to the UK, to Dundee. I then decided to embark on a PhD and work on Parkinson's disease and signaling transduction pathways in Parkinson's disease. So basically taking a gene that causes the disease and then unraveling the pathway step by step. So my research was...
Now I'm going to stop you because it's signaling. This is for laymen. So a signaling pathway is really a difficult word.
Okay.
So I think most of the other things is you went, you made a number of steps towards becoming not only a clinician, but also actually a neurologist, if I may say so, in Parkinson, but you're also into things like cellular pathways, and perhaps you can tell a bit better. You can probably tell better than me what that entails.
So
you can tell just about the, you were telling about the
do you want me to start again?
No, I want you to just move on and tell about the signaling pathway.
Okay, no, so basically I think what is maybe unique about myself is that I'm a clinician. So I do see patients in the hospital, I do clinical trials, but I'm also working mainly in a basic science laboratory with basic scientists. And this opens up amazing doors because I can talk to clinicians, I can talk to patients, and I can also talk to scientists. And often it is this interaction between all these different parties that is the difficult path to navigate. And my research interest and my clinical focus is on Parkinson's disease. So that's a neurodegenerative condition that affects mostly older people. But so I'm interested in the genetics of Parkinson's disease, and you will be surprised to learn that there are monogenic or genetic Parkinson's disease syndromes that can also affect very young people. So that's maybe my only connection towards pediatrics. And we are very interested here in Dundee in the lysosome, but coming from the Parkinson's perspective, because what we have learned, or where our research has taken us, is that by ways that we don't really understand as yet, that there's lysosomal dysfunction, that the lysosome does not function properly, is also at the heart of Parkinson's disease. And this has led us to basically come and work on the lysosome. So from probably a different direction than you working on metabolic diseases and lysosomal storage disorders.
Yeah, so I really like the way you perceive your role, and also like the thought of working together with basic scientists, clinicians, patients, and sort of together get a lot further than you would ever get on your own. Even with a massive group, it will still this translation is really important. So I see the difficult differences, but I also see the overlap, and perhaps So this is where Batten disease comes in, because it has some similarities with Parkinson's disease if you look at it from a perspective. If you get rid of the age, then a lot of the way they walk and also the problems in their heads are a bit like they have resemblance to Parkinson.
Absolutely.
Yeah. So go on. What happened after?
Okay, so we are a center of expertise. So we have quite a lot of research volume going on Parkinson's disease. And I work with a really, really famous biochemist. His name is Professor Dario Alessi. And he is really famous for all the work that he has done on a gene or protein that's called leucine-rich repeat kinase 2. LRK2. I know this is technical and I step away from the technicalities, but basically he's been working on this for the last 20 years. So genetic changes as a cause for Parkinson's disease and LRK2 were discovered in 2004, actually by someone who's now a really good friend of mine, Alexander Zimbrich. And, so it was then just known that LRK2 is a kinase, so a protein that puts phosphate groups on other proteins and thereby acts as a master regulator in a cell.
Okay,
but nothing.
Yeah, so the master regulator in cells, that with that, for the people that don't know biology, you can imagine that you have a lot of functions in your cell to make things work. But at sometimes the circumstances in the cell are different than in others, and it's as if you are in a car, you want to gas when you want to move forward, but you want to also use your brakes when you don't. And these kinds of decisions, they are actually there in cells as well. So this is the type of master regulator you're talking about, right?
So master regulator that tells other proteins, but just adding a phosphate group, switch yourself on, become active, become inactive, change your location in a cell, and so on. So it's really like a really common mechanism in cells. And it was really exciting that LRK2, a kinase, was involved in a neurodegenerative condition, so in Parkinson's disease, because what is quite interesting is that a lot of diseases have genetic changes in kinases, and kinases are potentially what we call druggable, which means that you can actually develop an inhibitor to a kinase and you can treat the disease. And that is the big question in neurodegenerative conditions, so Parkinson's disease, Alzheimer's disease, and I think that's also where the crossover to lysosomal storage disorders comes. Like, while we have an understanding of why, what happens and why the disease occurs, but we don't really have treatments. So basically,
I didn't know that they were druggable. So and now I'm checking whether I understand it well. The kinases, they are sort of the on-off buttons of proteins. If there's a phosphate group on, it may be active or inactive, and if it's off, it will reverse. And so, why are they druggable, and what would that do to potential treatments for Batten disease?
Okay. Well, maybe not Batten disease. So I'm now talking about diseases that have an underlying genetic change in a kinase. So in these enzymes or these proteins that are these master regulators in a cell. What you often see is, and that occurs in cancer, in diabetes, and with LRK2, the protein that we are working on in Parkinson's disease, that genetic changes result in a hyperactivation of this master regulator. And these druggable in terms of kinases means that you can develop specific inhibitors, so drugs that slow down and put a brake on these enzymes. And that's the mechanism. So there are over 70 approved, FDA-approved medications that specifically target kinases, like LRK2. And for example, now, for the last 2 or 3 years, there have been treatments targeting LRK2, so LRK2 inhibitors, for the treatment of Parkinson's disease in clinical trials. So these are all drugs that try to dampen down and inhibit the LRK2 kinase. And most of the
sorry, now and then I think I need a sort of gap to really understand what you're saying. But I was wondering about kinases and inhibiting them, even if it's possible with a drug. It feels a bit like you're using the brake all the time, or actually using the gas all the time. That sounds a bit scary as well.
yes, you need to somehow get, you need to get the balance right. But often what you see in these mutations, in genetic changes that affect diseases that are caused by kinase mutations, is that the activation, so that the mutation, the genetic change, results in an overactivation that is like 10-fold, 50-fold, 100 or 1,000-fold. And and then you basically need to somehow just bring it down to a normal level. And and that basically is the principle of treatment for lots of different diseases in cancer and diabetes, but it's not something that that has been done as yet in in neurodegenerative condition. And LRK2 mutations, so genetic changes in LRK2 that result in Parkinson's disease, the understood mechanism is that it's overactivation of this enzyme, and now there are clinical trials that test whether inhibiting, slowing down the activity of this LRK2 enzyme, whether this would help with the modifying or changing the progression of the disease. So that's not been proven. It's not an approved treatment, but it is something that is currently being tested in large international multi-site trials. And we are participating in these trials as well. But why I'm talking about this is twofold. A lot of the underlying biology to really understand, what's going on in LRK2 biology has been discovered here in Dundee by my colleague, Professor Dario Alessi. So without really the underlying biology and unraveling what goes wrong step by step in the LRK2 kinase pathway, without his work, we wouldn't really be at a stage where we would be treating, have these new medications in clinical trials targeting LRK2. And that's one thing. But then, and that's very exciting, but the verdict of whether that will help is still out there because the clinical trials are still ongoing.
I can imagine it's exciting, but it's probably not so much Batten related, or do you see?
No, but now I come
there.
Yeah. Because, so we are really trying to understand the biology of what goes wrong in Parkinson's disease when LRK2 is involved. And that has really, over the last few years, has really implicated the lysosome in Parkinson's disease, and in particular in LRK2 related Parkinson's disease. So over the last years, we've become really interested in how can we study the lysosome. And our laboratory, we are not cell biologists. We do a lot of mass spectrometry, and we are biochemists. So what we had, what was basically playing on our mind was, how can we look at lysosomal changes in disease? And one of the problems that, that's our approach, how we looked at it, is if you imagine a cell, and the cell is, a cell is quite small, but, in the context of a lysosome is tiny. And if you look at how much lysosomes, if you take the whole content of lysosomes in a cell, lysosomes make maybe up to 1 to 3% of the volume of a cell's content. So it's a tiny fraction of a cell's content that is represented by the lysosome. So if you then look at the whole cell, and you want to look at lysosomal changes, what then happens is that you are faced with the problem of signal to noise. So there may be a signal in your cell, but this signal that's specific for the lysosome is diluted out and lost in the noise of the rest of the cell, in these 97% of the other content of a cell. So we've been thinking about how can we go about this and really zoom in on the lysosome. And then we've been very, very lucky that we've been working with other people in the field, who are really experts on the lysosome. And one of them is Professor Abu Ramayi at the University of Stanford in the United States. And he's developed very, very sophisticated methods of studying the lysosome. But what his system requires is that the lysosome is tagged with a foreign protein. So something that we can use to fish out the lysosomes from a cell. And that is possible, like when you work with cell lines or with other disease models, but obviously not in a human being, because humans have normal native lysosomes and not anything tagged on their lysosomes. So, but we have been using this kind of principle of purifying and really like isolating lysosomes as the underlying principle of how we go about to look at lysosomes and lysosomal biology and what goes wrong in lysosomes in clinical samples. And that is where then we come in, where we met, because I first approached you with the idea of taking this forward together.
Yeah, because the same professor from Stanford University, he described it just a couple of years ago that if you use this strategy, you can get these lysosomes out the way you so nicely described, and then look into these lysosomes. And he found out that there were really peculiar, really very striking differences in patients with Batten disease that suggest that the problem of having Batten disease is now much better understood, and it probably relates to getting these to stores. The thing that stores tells something about the problem that goes on, and we have a much better feel for that, at least with this fantastic technique that until now was not available for patients. So it could also only be used there. So this is indeed where you contacted us.
Exactly. So if I remember, like we've been doing a lot of work on this, like, optimizing the method and then trying it out in healthy individuals, and, really in pain-stricken work, we've been able to somehow show that if I take blood, so basically I take blood I take a lot of blood from my husband for these purposes with ethical permission, or from other people in the lab here, so non-clinical samples, so samples from healthy volunteers, where I take a small blood sample, like maybe like half a teaspoonful or a teaspoon, and then like I isolate cells, white blood cells from blood. That takes about 10-15 minutes. And then what we do is we have to crush those cells open because we need to release the content of these cells. And then we do a step where we basically, like, you have to imagine, like, a key and a lock system. So where we have an antibody that specifically fishes out the lysosome via a specific protein in the lysosomal membrane. And then, like, basically we are left with these enriched lysosomes on magnetic beads. And then, like, it takes further processing to then look at this by mass spectrometry or other biochemical ways of doing it. So by the time when I first reached out to you, actually the connection came also that we asked the NCL Foundation and other foundations who work, who know who are the best people in the world to work, clinical people to work on this condition. So we were put in contact, with yourself. And then, like, so we reached out, and then I think, like, it might have come across as a crazy idea when we told you, well, we have this idea that we can isolate lysosomes, and that will be able, and by that way, we will be able to look what goes wrong in the lysosomes of people with Batten disease. And the reason why now you could ask, I'm a Parkinson neurologist, so why do I not do that in Parkinson's disease? And why do I do it? Why is this
question? Good question. Yeah. Why?
But I think, like, if I talk to people who are Parkinson's experts, I need to really, like, make a case for why I believe that there's lysosomal dysfunction as an underlying disease-causing mechanism in Parkinson's disease. Whereas, if you want to make that case in a lysosomal storage disorder, it's much, 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 much clearer to people. So because we are working extensively with, with our joint colleague in Stanford, so Monta Aboramay, and because of his discovery that he has discovered what's the specific metabolite, so what accumulates in the lysosome of CLN3 Batten, in the CLN3 Batten disease. So we had a starting point. We thought, what Monta had done, and that was published, I think, in a really high impact publication. So he had cells and he had other models, but not humans, where he showed that if you don't have a functional CLN3 or Batten protein, then like these metabolites can't shuttle out of the lysosome, they accumulate, and that probably causes the problems in this disease. So we said the best possible way to show that our new methodology of isolating lysosomes is valid and has utility in the clinical setting was to say, why don't we try to show what Monta had shown in other settings, in cells, why don't we try and show that in humans, in human blood from individuals with Batten disease. So that was the idea behind it, that might be the best possible disease model to validate our method.
And was it? Was it a good idea?
So it took quite a long time because when you work with patient samples, there is a lot of regulation and governance that needs to be put in place. So you need to write a protocol and you need to get ethical approval. So all of this really takes time. And once this was in place, we then visited you and another site in Germany, and what we did is you provided us fresh blood from some of your patients with Batten disease, and additionally we had also healthy controls, and then we did our procedure in your lab in Utrecht. So we brought everything what we needed to Utrecht and isolated lysosomes. And then what was really amazing, it took then quite a lot of time because we then had to freeze, these biochemical metabolites, the result of this assay, it needs to be stored at -80 degrees. You can't just put it in the post to Dundee or to Stanford. We had to keep it frozen on liquid, on dry ice.
-80 is really
-80 degrees, that's really cold. And it was horrendously expensive to ship the samples because it needed to be temperature controlled. We needed to ensure that that there was not a breakdown in the cooling chain. So the samples came then to our lab here in Dundee, so where I am at the moment. Then it required further processing, and then some of the analysis was done here in Dundee. And the mainstay of the analysis was actually done at the University of Stanford. So once we did further processing of the samples, we then had to again ship the samples on dry ice without breakdown of the cooling chain from the UK to the west coast of the United States. And that was where the samples were then analyzed by extremely sophisticated high-end mass spectrometry by Monta Aburama's lab. And what we saw, and that is really phenomenal. So if you looked at the whole blood, at these blood cells from the healthy controls and the patients with Batten disease, you didn't see a difference in these specific metabolites that Monta had identified as really critical in Batten disease. But when you then looked at the enriched lysosomes of the patients, or the controls versus the patients, you saw an up to 1,000-fold difference.
It's amazing, right? It's still amazing, and I think people are going to do the math now, and they're going to say, well, you have this 3%. So if there's 3%, then it cannot be 1,000-fold change, because then you would have a two times change or a 3 times change in the remainder. Or is it so? Where's the fault in this general thinking?
Well, no. I think it's just about what I talked about at the beginning. It's all about the signal to noise. So the difference in the lysosome is there. But if there's so much else going on around it, this difference is being leveled out. But then if you look in the right compartment in the lysosome, that's really only making up a fraction of a cell. If you look there, then you can suddenly see the difference. So shining the light and zooming in on the lysosome, I think that is probably the bottom line of this really hard research that we've done together over the last I don't know.
But it sounds like actually, I think I like you explaining also the niceness, but also the difficulties that you have to get through when doing this type of research, right? Because it's really Yeah, I don't come up with a name, but there's probably some Greek mythology example.
Sisyphus comes to mind.
Yeah,
and I think it takes, but I think I'm here in Dundee in this environment where kind of we take charge at the problem, and we are like bulldogs. We don't let go until we are there, until we found a solution. And that's a very privileged situation to be in, because research funding is often very, very short-lived, so you're always expected to have a result immediately. But actually, the really big discoveries do take a little bit longer and often come by chance.
Yeah, that's something that's called serendipity. I will I will get this out, but I have to take up my phone because I'm also on call. So I want to make sure that I'm not missing things that are really important. That's my colleague. So, and he doesn't usually call for nothing. So.
No, that's fine.
Hey Klaas. Ja, goed, ik ben aan het interviewen, maar ik dacht, er is vast iets aan de hand, dus ik dacht, ik neem op de foto. Ja. Lekker, ach die armen. Ja, nee, maar dat is helemaal oké. Ik ben nu met Esther Sammler uit Dundee aan het een gesprek aan het voeren. Dat is leuk. Dus ik vermaak me wel. En ik ga nu de komende uren kijken wat ik nog meer ga doen. Maar ik heb nog 1 interview hierna te gaan. Dus sowieso ben ik tot 11 uur, een uur of 1 onder de pan. En daarna moet ik maar eens gaan kijken wat er gaat gebeuren. Ja. Nee, ja, dat had ik gehoopt, maar die zijn de ver weg, dat lukte even niet praktisch. Dus die heb ik nog niet. Maar het gaat goed komen. Ik ga want er is er genoeg om te doen aan awareness, ook op andere manieren. Dus het komt goed. Ja, nou, dank voor het bellen. Ja. Ja, nou, nee, dat weet ik ook niet. Maar we spreken elkaar morgen. Oké. Hey, thanks. Bye. So we're in the middle of the challenge. So actually, it was my colleague asking how things are going.
So you should actually keep it in. I can understand a little bit, but
it's a sort of funny, right? So this is I don't pretend to be sort of to have an official term for it. It's not my new work, but it's nice to do. So.
We could also make this a talk about alternative careers.
Yeah, I want to get back to the Sisyphus, or at least to the painstaking that you're taking, but also it has to do with some gut feeling that you think I'm on the right track. This is really worth it, right? Yeah,
but I think there's also another element of a true heroism and the true heroes, because you have to imagine that all the people who have donated blood for us, all my colleagues here in the lab, my family. I think when my parents come from Germany, they usually give a sample because obviously they're older, and and sometimes I want to look at age-related changes. So so I think I'm ever so grateful to all these people who have made it possible that we can actually work with human samples. But then I think the true heroes are the patients with Batten disease who have participated. And also I think it's a huge sign of trust, that when I imagine I was the parent of these children, that that these families allow us to involve their children in research. And I think without this altruism and willingness to help, we wouldn't be able to make progress, because at the outset we were not able to somehow promise or tell them we will make any meaningful discovery. So that was a huge leap of faith on their behalf as well. So I'm always like very, very grateful to people who participate in research.
Yeah, that's really good to stress that. I was thinking that based on what you're telling, you have some. There's we keep the Greek path on. There's something we call a Tantalus torture. Have you ever heard of that?
No.
So it is. So it's a torture about that you can almost smell it or taste it, but you don't get it. I don't know the exact thing, but it's something like that. It's really a torture because it seems so close, and you're never going to get it. Is this so difficult that you can never bring it into practice?
That's a very good question. So I think the method, so what we've been doing, is not something that is easily to roll out, in clinical practice, because you do need, it's really very technical. Lots of different steps are involved, and there is just so much that can go wrong in terms of technicalities. But I think it is a start. So my theory is, we go through all this trouble to do all these like complex processing of these samples. But when you then know what you're looking for, I just wonder whether once you know what you're looking for, whether you can then also like deconstruct and make, and look at a different sample that's maybe not so complex. So I think it's always this is the discovery phase. And, now we need to do more research and, look at more individuals and really identify and find out what is the true, the true utility and impact of this kind of methodology in Batten disease, in different types of Batten disease. Does it allow us to track disease progression and so on? And also if there was a treatment, would it be possible to use this as a readout for potential treatment? So I think, I do think this is not something that can easily be rolled out to lots of different sites. It requires a lot of expertise, a lot of room for failure. But I do think you do need to go through this discovery in order to then develop tests.
Yeah, and you don't. So on one hand, you could say this is a disappointing side of it. But I was. You don't feel like that. I'm still not so totally surprised, not totally convinced that if you look at like how computers were built, once they were very large and now they're much smaller. So there could also be, once you know the path, you could probably make the path easier and get there in a maybe even smarter way. And now you found a way to accomplish something really incredible. But once you know the way, maybe there are less steps needed, or is that things that you find?
Oh no, I think so I think that a few things come to my mind. I think when you ask me can you roll this out in a larger scale? I mean, I think what I work a lot with clinical samples that have been collected from other sites. And even if you think it's a relatively easy to collect sample and there is not much technicality involved, you have no idea how many things can go wrong. And that is a lot what I do. I think what's the best sample, how do we store it, how do we collect it, so to somehow really get the most out of a sample that we can collect. So I think if we have agreed work streams and and there is you follow the same protocol and you know what the critical steps are, I think you can do it. And then I also think that if this was something that you wanted to integrate into clinical practice, in specialized centers, then I do think what you say with the analogy with the computer, you can automate these systems. So we already now work a lot with automated procedures where we we call it liquid handling platforms. But basically, like imagine where if I have to somehow do manually pipetting things, you can introduce a lot of error, or somebody comes and talks to me and I forget, oh, was I here in the 5th tube or in the 6th tube? But if you have a relatively easy mechanical robot that just does these repetitive tasks for you, you can achieve
the same time, exactly the same way every time. Yeah, then things will get both easier and also better.
Yeah.
Yeah.
I think like, then you need to somehow just like, you need the development and then like, also like the foolproofing of the systems. But I think like, where I see myself is more probably at the discovery phase. And then I think like, it takes other people whose expertise is to really streamline and implement this in
But I'm glad to hear that there, even if you're not going to do it, that there may be some avenues to really make this clinical. And now I was thinking a bit laterally, what other diseases could benefit from this? Is that I mean, it seems obvious that other lysosomal storage disorders could benefit from this. Is that as obvious as it seems, or are there loopholes in that thought?
Well, no, I do think like other lysosomal storage disorders, then but again, like then coming back to what I'm working on. So I work in on the other spectrum, in the older populations, on Parkinson's disease and neurodegenerative conditions. So we have been very, very fortunate that we are working, so a charity for Parkinson's disease is the Michael J. Fox Foundation in the States. So we've been very, very fortunate that we have received funding from them, and we will use our lysosomal, this lysosomal lyso-IP technology to really look at quite a large number of people with Parkinson's disease. People with Parkinson's disease without any genetic changes, so what we call idiopathic disease, where we don't know why the disease occurs, but then also in individuals with genetic changes such as LRK2. So I think there are lots of diseases where this might be really relevant.
Yeah, and do you anticipate that you would actually find these changes in lysosomal changes in both groups, or do you think it will be perhaps mostly the genetic ones and the other ones will have a different profile, or is it an open question?
I think it's an open question, but there's a lot of evidence that there's lysosomal dysfunction in Parkinson's disease in general, and then in certain genetic types even more so. But things that I wonder, where I think the verdict is out, is cognitive impairment, so memory problems and dementia. Could you ask the question, is if you have like cognitive impairment, is that because of lysosomal dysfunction? Because in Parkinson's disease, you have individuals who have just mainly motor symptoms, and then you have people with Parkinson's disease and other types of Parkinsonism who have motor symptoms of Parkinson's disease and dementia. It would be really, really interesting to know whether there was a difference at the level of the lysosome between the normal, typical Parkinson's disease that is mainly motor signs, and then the Parkinson's disease that has this cognitive impairment and dementia component to it.
Yeah.
So that's these are things that I'm very, very interested in. And then does the lysosomal dysfunction differ between genetic Parkinson's disease and idiopathic Parkinson's disease, or different types of genetic Parkinson's? Because you already mentioned this, but another link of Parkinson's disease with the lysosome is that there are lots of components that are part of the lysosomal machinery can cause, if there's something wrong, like if there are mutations, can cause Parkinson's disease.
Yeah, it's known that some actually, if you're a carrier, you may have an increased risk for some lysosomal disorders for Parkinson. Yeah, I'm aware of that. I want to discuss with you a few more minutes about the question how to get more research done, or to move faster in terms of getting a treatment for Batten disease, or at least understanding it even better. I've been talking to this is a part of my 24-hour challenge, is how do you increase awareness, and can we think of new ways to make things happen even when the circumstances are the same as they are now. So, and one of them would be how to create more awareness or to create more funding to make this happen. From your perspective, could you share some thoughts or advice about how to make that? Or
yeah, so as part of my of my role as a clinician and a scientist in a basic science laboratory, I think the communication and the outreach to the public and is really important, because people need to understand why there's a need for more research. People need to understand why basic science is quite costly and why tax money needs to be spent for science. So what we do a lot is we have a Parkinson's disease research interest group. So these are individuals who are somehow affected by Parkinson's, either because they have a diagnosis and are living with the condition, or they have family members with the condition, or their spouse, or a friend has Parkinson's. So we have a really hardcore group of people who work with us, and it's really fascinating. So they come, we meet every couple of months here in our institute. It's a group of about 30 individuals, and we then usually have plans of how can we increase awareness of Parkinson's. And the way we do that is we've organized webinars, where we invited really leading people in the Parkinson field from the States to talk to us. We had a Nobel laureate come to Edinburgh and give a lecture in the Royal College of Physicians, which is an amazing building. And by means of helping of Parkinson's disease charities here in the UK, helping us to raise awareness, to reach out to their members, we've been able to attract a lot of attention and bringing, I sometimes have, we sometimes have 100 people coming to open lab days here to our Basic Science Institute, and we would have so many more people, but it needs to be, we are restricted by how many people we can look after.
So it was not, there were more than 100 that wanted it, but not everybody could come. Yeah.
No, 100 people in the building.
Yeah.
So, and we have, and then in the Royal College of Physicians in Edinburgh, which is an amazing building. And, we organized for the first description of the assay of the shaking palsy published by James Parkinson's disease over 200 years ago. We had the original leaflet or, description exhibited when people with Parkinson's came and saw that. So yeah, so I think that is really important, because you do need the buy-in from people who are affected by the condition to participate in research and in clinical trials. But then it's also really important to reach the wider public, because if it's out of out of mind, out of kind of you need to somehow...
if you don't think about it, it becomes less important, something like that, right?
Exactly.
Yeah, and I think that is probably this. I would have thought it would be easy for Parkinson to get money, because it's such a frequent disorder as compared to metabolic diseases. So it's interesting to hear that even for Parkinson's disease, it's still something you have to be aware of and
I think
do
it's all a matter of perspective. When you look at how much money goes into diabetes or in Alzheimer's disease, so, or in cancer, I think Parkinson's disease
this is small
amongst these diseases is still the orphan disease. But but this is then even completely different for the kind of conditions that you work on. But I think there I could imagine that you have probably your foundations and your charities will be incredibly powerful. And I could imagine that also the families who are affected by these terrible conditions are are the best advocates to raise awareness for the condition and also for the need for money. And luckily, there are also like, recognition that orphan diseases, so really ultra rare diseases need to be treated differently in terms of funding and help to progress research than these other much more common conditions.
Yeah. Okay, I think that's a wise thing to say. I get the impression that many of the parents are often overwhelmed by the amount of work they have to do to spend on their children that are so much deteriorating. That makes them being voicing the importance quite a task. But I do think it's good to be aware that those are the ones that have to talk about this. I think I'm going to end the conversation. It was really nice having this discussion, and we'll speak at some other time soon.
Okay, thank you so much for inviting me, and good luck with your challenge.
Thank you.